Okay, so welcome back to our metabolism and enzyme part 3, which is in your Form 4 Biology Chapter 4. So in today's video, we are going to talk about what are the factors that affect our enzymes activity and what are the application of enzymes in our daily life. So first, start from the mechanisms of enzyme and also what are the factors that are going to affect our enzyme. So first factor that affect the enzymes is temperature. So when the temperature rises, the kinetic energy of the substrate and also enzyme, they will increase. Okay, so the enzyme and substrate molecules, they will move faster. So this is going to increase the frequency of the effective collision, which they will collide more frequently and between the substrate and enzyme molecules. So when the frequency of the effective collision increase, the rate of reaction between enzyme and substrates also increase. So the optimum temperature when the uh, when both of this enzyme and also substrates they are at their optimal temperature, the enzymes reaction is at their maximum. Okay, and the optimal temperature for enzymes in the human body is 37 degrees Celsius. That's why our body temperature without a fever normally will be always kept constant around 37 degrees Celsius. So after reaching the optimal temperature, so any further increase in temperature will reduce. All right, later I will show a graph. You can see how is the reaction going. So when the temperature increase very high, the enzyme activity actually get reduced. This is because all the enzymes they get denatured. Denatured is where they are getting destroyed. It's a term for like protein. So because enzyme is a type of protein, so when the enzymes are at a very high temperature, they will get denatured and their activity reduce. So you can see when this is the x-axis for the temperature. So when the enzymes they are getting in a when the temperature increase and the enzymes they will increase in the rate of reactions, and when they achieve their optimum temperature, they are going to be at their maximum activity. All right, and then after further increase of the temperature is going to denature the enzyme, and this is going to decrease the enzyme activity. Okay, so remember the progression of this graph. So the next factor that going to affect our enzyme is the effects of pH. So pH is the balance between our acid and also our bases or alkali. So most enzymes they are most active in the range of between 6 and 8. So in our human body is around 7. Okay. So for example, like the salivary amylase, which you can find this enzyme in your saliva, and the function is to digest your carbohydrates. Okay, if you forget about that. So however, there are some exception um, for the pH of the enzyme in our body. For example, like pepsin. Pepsin, you can find this in our stomach to digest the protein. Okay, so the pepsin, because is found in the stomach, so our stomach is in a more acidic environment. So the optimal pH for pepsin enzyme is around 1.5 to 2.5, which is a acidic environment. And in sorry, in another hand, trypsin enzyme, which is found in the duodenum, they only works well in an alkaline medium, right? So in the duodenum, they works well in the alkaline medium. So trypsin, the optimum pH for it is around 8.5, which is slightly alkaline. But for most of the enzyme, it's going to be around 6 to 8, which is quite neutral for the pH, okay? So why is there a change of enzyme activity when there is a change in pH value? This is because when there's a change in pH value, they're going to change the charge, okay? So the ion H plus. So you know that the more H plus we have, the more acidic the medium is going to be. So the change in pH is going to change the amount of the H plus ions at the active site. So when the H plus ions is being affected, and then the enzyme uh, and substrate complex, they cannot be formed, all right? So this is why it's always important for us to keep our uh, pH body in the optimum levels. Like for example, in the pH 7 for our mouth and in the stomach in the acidic pH. Okay, and this is the graph. You can see This is the x-axis for the pH. So between 6 and 8, which is the optimum pH for the salivary amylase or most of the enzyme in the human body. And then for the enzyme pepsin, is in a more acidic pH. And then for the enzyme trypsin, which is in your duodenum, in your small intestine, is a slightly alkali pH. Okay. And you can see this is a folded protein. So when they get denatured, this the normal protein, which is a normal enzyme, they are in a 3D configuration or in a 3D shape. But when their pH or even their temperature changes, they're going to get denatured and they're going to become this unfolded protein. Okay, it's important for you to know that when the pH changes and when the protein get when the enzymes get denatured, it can actually be reversible. It means that after the pH becomes normal back, and then the pH is going to becomes when the pH becomes normal back, and the enzyme is going to becomes normal back as well, and the enzyme is going to work like normal. But when the temperature increase to a very high temperature, and we get a denatured protein or a denatured enzyme. So when you get back to a normal temperature, your enzyme is no longer function like normal. Okay, so that is the difference between the factor temperature and also pH. Okay, so the next factor we want to talk about is the effects of the substrate concentration. So when the substrate concentration increases, the effective collisions between the substrate and enzymes are going to increase as well. But they're going to increase until a certain maximum level, and then the rate of reaction going to stay constant. I will show you the graph later as well. So in this case, at the maximum level, although we further increase our substrate concentration, but due to the limited amount of the enzyme, the enzymes here is going to become a limiting factor. So that's why the whole reaction is going to be constant. So you can see this. This is the x-axis for the concentration of substrate. So when the concentration of substrate increase, the activity increase, but it's going to become constant. So in a straight line. This is because all of the substrates, all of the substrates is engaged with the limited number of the enzyme. The light yellow color is the enzyme. And although we have a lot of the extra substrate, but there's no enzyme to fit the, the substrate, all right? There's no enzyme to feed the substrate. The enzyme is saturated. So the enzyme here becomes a limiting factor. So that's why the rate of reaction becomes constant, all right? And then the next and also the last factor to affect our enzyme activity, of course, is the enzyme concentration. So same like the substrate concentration, when the concentration of enzyme increases, and the rate is going to increase as well because there are more active sites on the enzyme for the reaction. Okay, but in this case, at the maximum level, the rate of reaction is going to become constant as well. So this is because the substrate is going to become the limiting factors here. Although we give a lots of the enzymes in, but there are limited number of the substrates. Okay, so the substrate becomes a limiting factor. So this is like opposite when compared to the last factor, the substrate concentration. Okay. So this is a graph for this. See, although we increase the enzyme, so the rate of reaction is going to increase as well, but it's going to reach a constant level. And the substrate is going to become the limiting factor. So lastly, we're going to talk a little bit on the applications of enzymes in our daily activity. So for example, we have some digestive enzyme that is used in a medical center or can be given as a supplement for those who are having a problem with digestion. And also in some milk, we use a lactose enzyme. Still remember how we name our enzyme, the nomenclature. So lactose enzyme also can be named as lactase, ASE, okay? So can be used in lactose-free milk. For those who are having a problem, we call it as a lactose intolerance. Then we can have this lactose-free milk by adding this lactase in. And also we can have protease, which digest your protein to separate the fish skin. And also we can have other types of the enzymes in our biodetergent, which help us to digest a lot of the chemicals or molecules in our daily life. Okay. So to recap for our video today, we actually talk about the 4 factors that affect our enzyme activity. So what are the 4 factors? We have the temperature, and we have the pH, and the most optimum, most of the enzyme, they will have an optimum pH of 7, around 6 to 8 in the human body. And the third factor is the substrate concentration. And the fourth one is the enzyme concentration. And lastly, we also talk about some applications of the enzymes in our daily life. For example, like the use in the medical sector or use as a supplementation for those having a digestive problem. All right. So that's all for today's video. Thank you very much and see you.